Hey guys, closing in on it. This is the last two lectures, this one and the one that you get on Friday. Be the last two lectures we have that are straight up new information. Everything else after that will be a review. So this part AP doesn't hit as heavily, so I've trimmed down the PowerPoints quite a bit. We need to know about bacterial transformation, electrophoresis, PCR, and DNA sequencing. And so that leaves out microarrays, it leaves out CRISPR, it leaves all kinds of stuff out. I'll still hit CRISPR a little bit probably today just because I can't help myself. So we're living in a world now where genetic modification and gene therapies and genetic identification, these are all becoming commonplace. We've got genetically modified foods, organ donors, stunts, and of course we've got 23andMe or organizations like this that can look at parts of your DNA. And again, we talked about that before. They're looking at your SNPs, not necessarily your sequences, but we do have the ability to sequence DNA. We can synthesize biological molecules using bacteria. This is humulin, which is human insulin produced by Eli Lilly from E. coli bacteria. And of course, we can use DNA to identify individuals. We can use it for a lot of different things. So let's look at the tools that we need to use some of these technologies. Tool number one, so we'll have just a few tools in this little toolbox, and it won't cover everything that all the technologies use, but it's a good starting place. And this stuff is used a lot for bacterial transformation, which is a kind of a complicated process that we need to get your heads around. All right, so an endonuclease, and I've brought this up a couple of times before. Exonucleases, remember, digest nucleic acids from the 5' end. And that's how we recycle, the excess nucleic acids that we produce in our bodies, mostly RNAs and things like that. It's also a way that we can break down viruses, things like that. But an endonuclease works a little bit differently. It cuts in the middle of a sequence of nucleotides. So, and it will cut at specific sequences. And they're called restriction endonucleases because they restrict what DNA is allowed to be in the cell. So these are originally isolated in bacteria. We find a lot of these in bacteria. So it's a protein that will cut a very specific sequence of DNA. So they don't want to cut their own chromosome. They'll have a couple of strategies to prevent that. One, they just won't have very many locations in the genome that have the sequence that the restriction enzyme cuts, sometimes none. Or they can also just stick a protein on that sequence that blocks the restriction enzyme from cutting that DNA. So their DNA is safe, but any DNA that comes in with that particular sequence gets cut. Almost every restriction enzyme cuts at a sequence of DNA that is like these phrases. Hopefully you've noticed what they are. And of course, my favorite one's this last one here. But these are palindromes. Now, when we have a palindrome in DNA, because it's anti-parallel, what we have is, instead of running the same way forwards and backwards like CATTAC, it's a palindrome that runs opposite directions on each strand. So it'll be read one way on one strand, and you will be able to read it again on the other strands. So here's a good example, and this is a very common one, actually. There are several restriction enzymes that cut at this particular site, or one very similar to it. But this GAATTC. So if this is 5' and 3', then this would be 5' and 3'. And so when you read this 5' to 3', you read GAATTC. And when you read this one, 5' to 3', GAATTC. So that's our restriction site. That's the place that gets cut by a restriction enzyme. It's called a restriction site. And this happens to be the restriction site for a particular enzyme called ECHO-R1. ECHO because it's E. coli, and R1 because it's the first restriction enzyme we identified from E. coli. So like I said before, they protect themselves from their own endonucleases by not having these restriction sites or by blocking these restriction sites with proteins. Usually they're short, 4 to 6 base pair sequences, small because it makes it more likely to just randomly appear in some sequence of DNA. So it makes it more likely. If you had like a 3,000 base pair long stretch of DNA that the restriction enzyme identified, then the restriction enzyme would almost never find that in any particular stretch of DNA, and any viral DNA, for example. But if it's short, just the random arrangement of base pairs means that this might appear sometimes. And so the restriction enzyme is going to cut those and thus protect the cell. We have a lot of these that we've isolated from different bacteria, EchoR1, HindD3, BAMH1. What's super cool about restriction enzymes for us, because we want to use this thing as a tool, right? We want to be able to cut DNA so we can put it back together and make new combinations of DNA. This is what we do with genetic technology. And so when it cuts, this restriction, so EchoR1, for example, will cut like this, and it leaves these sticky ends. So this A wants to base pair, right? This A right here, this A right here, and both these T's, they all want to base pair with another T, T, A sequence. So once I cut those, I can take another piece of DNA that I've cut with the same restriction enzyme, and I can patch it in there, and it will go in both ends. So I can splice a piece of DNA into the middle of another piece of DNA. So I can make recombinant DNA, DNA with more than one source. Okay, so we'll talk more about how we use that restriction enzyme in a bit. Just as a side note, CRISPR is actually a restriction enzyme mechanism, but it's an especially cool one because it's programmable. So it's a restriction enzyme that you give a strand of RNA to, and it will find a sequence of DNA that matches that stretch of RNA that you gave it, and it will cut the DNA at that particular sequence. So you can artificially assemble a stretch of RNA that matches a particular gene that you want to modify, and the restriction enzyme will go and cut the DNA at that particular location. And that's pretty cool. You know, it's very useful because then what the cell's going to do, whenever a cell, whether it's eukaryotic or prokaryotic, if it's got a broken chromosome, the first thing it's going to do is try and fix that because that's problematic. And so the way it fixes that is it scrambles around and tries to find a sequence of DNA that is similar to what got broken. And then it will rebuild that stretch of DNA on either side of that break using that sequence that it found that matches what was damaged. So what you do is you flood the cell with a bunch of copies of DNA in the sequence that you want. You cut the chromosome using CRISPR. Your cell responds by trying to repair that, and it looks around for a piece of DNA, and there's all these copies of this gene that you want, and it'll take the gene that you cut and change it into the gene that you want. You could take a cell that carries the gene for cystic fibrosis. You could target that piece of DNA with the CRISPR, cut it at that location. The cell scrambles around trying to find a copy to replace it with, and it replaces it with the proper working version so that you no longer have cystic fibrosis in this cell. Now, this is not terribly useful yet for somebody who has cystic fibrosis because it's very hard to get it into all the cells that it would need to be in, but you could easily change a zygote or an egg using this. There's also another really kind of cool thing with CRISPR, cool and creepy. It's called gene drive, and we can actually splice into a chromosome not only the repaired version of a gene, but also the tools and machinery to repair the gene. So if I make an egg cell with this repair unit in it, fertilize it with any sperm. It doesn't matter if that cell has a working version or a non-working version of that, let's say that cystic fibrosis gene. When the sperm fertilizes the egg, the gene drive mechanism from the egg will repair the DNA sequence of the sperm. And when that person goes to have children, the same thing will happen to their children. So they're going to add in that gene drive mechanism to every piece of DNA that they encounter that has a similar sequence. So for generations on, that thing is gone now. We've repaired it in everyone after that, in every child they produce. It's not this, 1 in 4 recessive combination. It's all of them. So there's some interesting studies going on to make it so that mosquitoes can't carry malaria. But it's kind of creepy because that means anybody could genetically modify the human race, splice in to one gamete, make an offspring with that, and now everybody that comes from that, every descendant from that person will have this genetic change. And over generations, that could end up being a huge number of people. And if it's something that's seriously beneficial, then it may end up in an awful lot of people. Same could be done with organisms out in the wild. So it's a little bit creepy because there's definitely a mad scientist aspect to it that is entirely too easy and practical. If you listen to podcasts, Radiolab has a couple of excellent ones on CRISPR that talk about gene drive. Worth a listen to. All right, so I want to talk about bacterial transformation. I mean, really, that's where we're going with all of these tools. But if you remember back to this transforming principle, when we talked about Griffith, he had these mice that when you injected them with one particular type of bacteria, they died. You injected them with the other kind, and it was harmless. But if you heat killed and then mixed right? We had this transformation happen that turned the harmless one into the deadly one. And he called that the transforming principle. And then later, Oswald Avery figured out that that was DNA. So how do bacteria transform one another? What is the thing they're picking up besides just being DNA? Because they're not just picking up whole chromosomes. That would change the cell entirely. What they're picking up is called plasmids. Plasmids. And plasmids are small rings of DNA. So if you're a bacterium, you've got a big chromosome with most of your genes on, all those operons and everything. But then you also have little rings of DNA that may be in your cytoplasm as well. And these can be exchanged from one bacteria to another. They can be picked up from the environment. This happens a lot when bacteria are stressed. And so it's just a small ring of DNA, usually carrying some useful trait that is shareable amongst bacteria. A lot of our antibiotic resistance genes are carried by plasmids and so easily transferred from one bacteria to another. But there are other characteristics that come with plasmids. So bacteria do a thing called conjugation, where the two of them will hook up next to each other, run a little tube of cytoplasm, and exchange plasmids back and forth. It's a little like sex, but it's not for reproduction. It's just to exchange some DNA, and it adds variation. And the DNA they're exchanging is plasmids. Like I said, if a bacterium dies, then it's going to release its plasmids, right? The cell's going to rupture. And its plasmids will be out there, and nearby bacteria will pick it up. And this is what happened with the transforming principle in Griffith's experiment. But we can exploit these plasmids as a tool. And what's really cool, now that we got the restriction enzymes we just talked about, is we can cut anything we want into those plasmids and have the bacteria pick that up. So here's some words about plasmids. I don't know that you need all of them. I think I summarized it all right there. It is a thing called horizontal transfer, which horizontal transfer is something I would spend a lot more time talking about if we weren't under these weird circumstances. But it's worth looking into also. It's super cool. It's basically this idea that we get a lot of genes that go from one species to another, or from one organism to another. I think I've mentioned in class before, one of the genes that makes our placenta work so well and made placental mammals possible was actually stolen from a virus, right? We have genes in our nuclear DNA that came from our mitochondria and are no longer produced by our mitochondrial DNA. So genes move from organism to organism all the time. But this is just one example of it. So here we've got a bacterial cell. We're showing some plasmids in here and plasmids in the environment that can be picked up. Usually not a lot of genes on these things, but they tend to be useful because that's what allows them to propagate. So what we do to transform bacteria is we take a plasmid, which is just a ring of DNA, we cut it with a restriction enzyme, we take a gene from some organism, say humans. Let's say this is the gene for making insulin. We cut out that DNA. We got sticky ends on both ends of this because of the restriction enzyme we used. We got sticky ends on either side of the cut of the plasmid. Those ends stick together. Now we've got a new plasmid that has the gene for human insulin in it. We put that into bacteria. Basically, all I have to do is stress them, put them in a test tube with the plasmids, stress them, and they'll pick it up. What we always do every year for our bacterial transformation lab is we heat shock them. There's a little bit of a chemical component as well that makes them a little more receptive to plasmids, but we heat shock them, and that causes them to pick up the plasmids, and then they're transformed. So now I got a bacteria that has traits it didn't have before. This bacteria right here, this can make human insulin because it has the gene for human insulin in it. So this is just another diagram showing the same thing, but it shows the sticky ends. I've cut my desired gene out of this. I've cut my plasmid, and I spliced that into there. And now I've got my recombinant DNA, right? My recombinant plasmid. Again, recombinant just means DNA from two different sources. If this is, like I said, the gene for insulin, then this recombinant plasmid has DNA from humans and from bacteria. Now, one question. What enzyme are we going to need right there and right there? Because they stick it together with base pairs, but we didn't get the backbone stuck together. See if you remember from when we were talking about DNA replication, what enzyme you need to stick together the backbone of DNA. And that leads us to our third tool, which is ligase. Hopefully that was what you came up with. But when we cut it, and we've got those sticky ends, we're going to stick it together with this new piece of DNA. If we're going to get this connection made, these connections here, 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 and here all require ligase so that we can tie up those 5' 3' ends and get it to be one continuous stretch of DNA. So DNA ligase joins those strands together. Sometimes we say it seals the nicks. I'm not sure why they say that. So this allows us to take this piece of recombinant DNA, of course it would be a plasmid, not a strip like this, it would be a loop. And it allows us to have the bacteria produce a protein that's not a bacterial protein, right? We can produce human insulin, which is a protein, in E. coli. And that allows us to mass produce it. So instead of harvesting it from pigs or other humans or anything else, we can just have bacteria grow it in huge vats and have gallons of this stuff. I did skip a little step here. If I take human DNA, cut it right out of my own genome, and splice it into a bacterium, there's going to be a major problem. The bacteria is not going to be able to use that to make proper insulin. See if you can come up with a one-word answer as to why that won't work. So hopefully that one word is introns, right? My DNA, my code for insulin, has introns in it. And if I put that into bacteria do not remove introns. They don't have SNRPs. They don't have the mechanism to remove introns. They can't make a spliceosome. And so it's going to make this huge protein that's way too big and doesn't do what insulin does. Remember, we've got these exons and introns, and we don't use these when we make the RNA. But bacteria would use the entire sequence to make the RNA and therefore to make the protein. So the protein would be way too big and non-functional. Now let me ask you this. There is one enzyme that we've already talked about that would make it so that we don't have to worry about this anymore. See if you can think of that. Pause the video. I'll give you a hint, and you can pause the video again. It's in the virus unit we just talked about last week. Maybe you didn't come up with it. It only got a quick mention, but this enzyme is reverse transcriptase. All we have to do is take RNA, my mature transcripts for how I make insulin. That's the whole thing with no introns, right? So I could take that, reverse transcriptase that into DNA, cut the ends with restriction enzymes that leave sticky ends, splice that into a plasmid, put that into the bacterium, let them grow, and now we've got bacteria that produce proper, fully functioning, no introns, human insulin. Pretty cool trick. So big picture, the steps of using bacteria to mass produce insulin. And again, we've gone through this a couple of times. Take a plasmid, cut it with a restriction enzyme. We're going to take the mature transcript, use reverse transcriptase to make DNA, cut that with the restriction enzyme. Put those two cut pieces together to make a new recombinant plasmid, stress a bacteria so that it'll pick up that recombinant plasmid, and let the bacteria grow so you have millions and millions and millions of these cells all producing the desired protein. In this case, insulin. And this is exactly what Eli Lilly did when they made the first humulin. It's really not that easy to transform bacteria. When I've got a little vial of bacteria with millions of them in there, and I put in my plasmid and I heat shock them, right? A tiny percentage of those bacteria are actually going to pick up that piece of DNA. So I can't just take all the bacteria that are in that vial and grow it because most of them won't produce insulin. I only want the ones that do produce insulin. So on that plasmid, I add something else. I add our fifth tool here, marker genes. And a marker gene can be something as simple as antibiotic resistance. So I got a plasmid that has, let's just pick an antibiotic, say ampicillin resistance. That's the one we usually use in the lab. It has a gene that allows it to survive the presence of ampicillin, which most bacteria don't. I splice open that plasmid. I put in the human insulin gene. So now I've got a plasmid with the human insulin gene and the ampicillin resistance gene. I heat shock a bunch of bacteria so they pick this up. Now in that little test tube, almost none of them are going to pick it up, but the few that do can survive on a petri dish that has ampicillin in it. And the ones that don't, the ones that didn't pick up the plasmid, they can't survive. So I plate them on a plate with ampicillin, and the ones that grow, then I take those and put them in my big vat of beef broth because I know every one of those got the plasmid that I made. So they don't always have to be antibiotic resistant. Sometimes they're colorations. Sometimes the gene that we use to demonstrate transformation is a GFP, is a green fluorescing protein. It makes glowy bacteria, but that can be used as a marker gene too. So if you have a plasmid that has GFP and then you splice into it your desired gene, you know that if the bacteria glow, they're the ones you want. And so you only grow those. So marker genes are pretty important. So today we've covered bacterial transformation. We'll look at more of the DNA types of technologies. In the next one, we'll do PCR, gel electrophoresis, and sequencing. And then we'll be done. Still looking at a quiz next week. Shouldn't be too bad, short and sweet. If you know somebody who's in AP chemistry, that'll give you some idea what the test will be like. Small, multiple choice, that kind of thing. Hang in there. You're doing great. Love you guys.